Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Sandra Sanchez-Roige will present what genetics is telling us about substance use disorders. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 184 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded more than $400 million to fund more than 6,400 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Sandra Sanchez-Roige. Dr. Sanchez-Roige is an associate professor in the Department of Psychiatry at the University of California, San Diego, and the Department of Medicine, Division of Genetic Medicine at Vanderbilt University Medical Center. She was a 2018 BBRF Young Investigator. Our webinar will begin with her presentation, which will then be followed by Q&A. To submit your questions, please use the questions tab on the control panel on your screen. Please feel free to submit your questions at any time following the presentation. I'll ask as many questions as possible in the time allotted. And now I'm pleased to introduce Dr. Sanchez-Roige. Sandra, the floor is yours.
Thank you, Jeff. It is an absolute pleasure to be here. If you're listening, like me, you must have a profound interest in learning about substance use disorders. This talk is for anyone that shares this interest. If you are a scientist, I will walk you through the most recent advances in the field of substance use disorder genetics. If you are not a scientist, perhaps a doctor with a deep desire in improving treatment for those with substance use disorders, or perhaps you or a loved one are struggling with this condition, this talk will provide you a window into why should we be studying the genetics of substance use disorders, what have the studies so far uncovered, and where do we need to go to transform these discoveries into concrete actions, effective interventions, and bring hope to those in need. Substance use disorders have been the focus of all my career. Although this picture may represent a severe or extreme manifestation, substance use disorders are amongst the commonest psychiatric conditions. The etiology is complex because it is embedded in a web of environmental and genetic factors. And the biggest challenge that we're facing, and potentially the reason why you're here today, is because you're aware that prevention, diagnosis, and treatment mechanisms are only limited. Over the past 6 years, there has been an explosion of large studies aimed at finding the genetic factors that may be associated with substance use disorders, some of which we will be reviewing together today. These studies are vital because they can discover new biological mechanisms, lead to new interventions, and make a significant impact in people's lives. And this is a principle that truly fuels my research. It has been very inspiring to see the progress achieved in the field of human genetics. At the forefront of this progress have been genome-wide association studies, or GWAS, as you will persistently hear me say today. This methodology sounds very technical, but the principle is simple. GWAS are designed to mine or scan across our genome in the quest of identifying genomic regions that may be associated with a trait, for example, alcohol dependence. GWAS have proved to be extremely successful tools, as I hope you will agree with me at the end of the talk today, but they have a major shortcoming that I must also disclose before we go any further, which is in order to identify meaningful results, we need extremely large sample sizes. In order to scan our genome, we also need sophisticated machines that over the years have also become more inexpensive to use. And this reduction in cost was really good news because it allowed the possibility of genotyping thousands to millions of people, which again is necessary to have sufficient power for gene discovery. Computational approaches and methods to analyze these vast amounts of data have also become more refined over the years, but what I want to emphasize today is to think deeply about the phenotype. The biology that we're able to extract is heavily dependent on the trait and the population that we choose to perform our study. So how do we measure substance use disorders? When we go to the doctor, they can very accurately measure our blood pressure, for example. They have very precise instruments for that. But when we go to a psychiatrist or a psychologist's office, quite likely we would not be able to spot any single piece of equipment except for one book, the Diagnostic and Statistical Manual of Mental Disorders, or DSM. It contains diagnostic criteria for all mental disorders. And they would ask us a bunch of questions to determine whether we meet certain criteria, and if we carry two or more of the symptoms that we can see in the screen, we would be diagnosed with a substance use disorder. So in this sense, this trait, DSM diagnosis, could serve as our gold standard. We could ascertain individuals diagnosed with an alcohol use disorder and perform a GWAS. These have indeed been the major efforts led by the largest international consortium on psychiatric genetics, the PGC Substance Use Disorder Work Group, of which I am honored to be a member along with many others. And one of the landmark papers by this group back in 2018, and the first study that I will be presenting today, was focused on assembling multiple cohorts to perform a GWAS of alcohol dependence as defined by the DSM. And here we have the GWAS results summarized in a single plot. Each dot represents a genetic variant or a single nucleotide polymorphism, or SNP, spread along the chromosomes on the horizontal axis, and the strength of the association between a SNP and a trait, in this case alcohol dependence, is plotted on the vertical axis. These plots are commonly called Manhattan plots because scientists expect that their analysis will be so successful that we will be able to see many towers or dots surpassing this significant threshold red line, resembling the skyline of the New York City. But we can see that after so much rigor and effort, this is more akin to a Washington plot, as this study was able to reveal only one locus, including one of the genes that regulates how ethanol is metabolized in the body. So it became evident that substance use disorders, like all complex traits, are highly polygenic, meaning that thousands of genetic variants, each with small effect sizes, confer risk, and that much larger sample sizes would be needed to boost gene discovery. But it is not hard to imagine that these traditional study designs, which are drawn from case control designs, come at a cost, because deeply characterizing individuals is expensive, slow. We may also be including high levels of heterogeneity. For example, there are over 2,000 unique combinations to receive a substance use disorder diagnosis. We may also have problems with misclassification, because we have to draw a line of separation between these two groups, which is artificial, and that can have an impact on statistical power. To complicate things even further, substance use disorders develop in a chronological fashion, from experimental to regular use, compulsive use, cessation, and, as it is often the case, relapse. And when we're using a binary case control study design, we're obscuring the signal that may be relevant at each of these stages, which could have enormous therapeutic value. It is possible that different genes may impact or have a different role at different stages. Complementary approach, and the one that I will be primarily focusing on today, is the use of a continuous or dimensional phenotypes. For example, our ability to self-regulate behavior or levels of alcohol consumption. And because they represent domains of normal function, they can be measured in the general population, even in online settings, thereby reaching very large sample sizes, which again are very much needed for genetic studies. So it is, of course, a trade-off or a compromise between obtaining a large sample size at the expense of phenotypic depth. In this talk, I will argue the value of both, but I will also emphasize the power of these small blue figurines. Back in 2016, I joined Dr. Abraham's Palmer laboratory as a postdoc, and the challenge that we wanted to solve was, where can we get high volumes of data that may be relevant to substance use disorders? And the answer was 23andMe. The beauty of working with a genetic company like 23andMe is that they have already genotyped millions of people. And so collecting phenotypic data could be targeted at large scale for a fraction of a cost. So we partnered with 23andMe, more specifically, we paid them to deploy an online survey that we compiled with input from psychologists, and our survey included well-established questionnaires that each captured different aspects of substance use involvement. And one of the phenotypes that we included in this survey, and the second study that I will be discussing today, are the scores from a 10-item questionnaire audit that measures past year alcohol use. We collected 25,000 responses from 23andMe research participants, which we combined with additional audit data from another population-based cohort, the UK Biobank. The UK Biobank is an academic initiative that has completely revolutionized the field of psychiatric genetics by providing genotype and phenotype data in 500,000 participants from the UK. We combined the data from these two data sets and performed a GWAS of audit, which uncovered 10 loci, 9 more than the inaugural GWAS of alcohol dependence that I showed in an earlier slide, including the familial ethanol metabolizing enzyme gene. Now, the beauty of this questionnaire audit is that it can dissect alcohol use from misuse. For example, the first 3 items measure aspects of consumption, such as the frequency of drinking, quantity of drinking, and patterns of binge drinking, that is, drinking too much too fast, which we named Audit C. And the remaining items measure problematic consequences of alcohol use, which we named Audit B. We performed 2 separate GWAS of these audit dimensions, Audit C at the top, Audit P at the bottom. And what was clear was that some loci, for example, again, ethanol metabolizing enzyme genes on chromosome 4, were consistent, but that the overlap was incomplete, suggesting that the genetic architecture of alcohol use and misuse is distinct, and emphasizing the value of deconstructing such 2 core symptoms of alcohol use disorder. We next wanted to examine how closely related these audit phenotypes were to clinically defined alcohol dependence. And to this aim, we used what it has been quite a revolutionary statistical method to perform genetic correlations. The uniqueness of this method is that, unlike phenotypic correlations, which are performed using the same individuals, genetic correlations can be performed across pairs of traits that are measured in independent cohorts. And that has many benefits. First, it is not necessary to measure all traits that you'd like to measure in the same cohort, which could be very expensive. And second, we're also partially removing effects of the environment because we're not using the same individuals and therefore they do not share the same environment. So we performed genetic correlations between audits and alcohol dependence, and we identified very strong, significant genetic correlations between these traits. And these findings are, to my view, extremely important because they suggest that we could combine clinical and dimensional phenotypes, such as alcohol-related traits from population-based cohorts, which are cheap and fast to measure, reaching unprecedented sample sizes, as beautifully illustrated by our recent work, thanks to the generous donations of the Brain and Behavior Research Foundation to Dr. Hang Zhao, whereby combining clinical data with audit has propelled the identification of 110 risk loci associated with problematic alcohol use, which is a dramatic increase from our original study back in 2018. The Brain and Behavior Research Foundation wrote a very accessible article summarizing this work, which I greatly enjoyed, and I leave a QR code for anyone interested in reading more. So overall, the projects that I have presented so far have considered complementary study designs. Using audits, we have begun to relatively inexpensively annotate different addiction stages. We have shown that alcohol use and misuse have a different genetic basis, and we have uncovered hundreds of novel genes associated with problematic alcohol use. We have also learned something that is very important to communicate, which is that alcohol use disorders, like all complex traits, are not single gene disorders, and that hundreds to thousands of genetic variants, each with small effect sizes, confer risk. We have also been reminded of the importance of data sharing and collaboration, both through international consortia, but also by involving people in research, which is one of the very few ways to be able to compile such critically large data sets, which, as I have hopefully convincingly illustrated, are much needed for genetic studies. The third study that I would like to present today pertains to aspects of prescription opioid use. The metrics for the current opioid epidemic in the US are shocking. Almost 130 people die every day in the US from opioid overdose. Every day. And whereas the path to opioid use disorder initiated with heroin in the early 1960s, by 2013, the majority of opioid users initiated with prescription pain relievers. And because prescription opioids are widespread in medical settings, we believe that this could be a phenotype that we could also capture in a population such as 23andMe. So in 2018, we extended our initial survey to 125,000 people. And one of the questions from the survey pertained to taking prescription painkillers not as prescribed. To our surprise, 20% of the 23andMe research participants reported having ever taken opioids not as prescribed. So we wondered if we could show that a simple question such as this one, which may capture how people misuse opioids, if a trait like this one measured in 23andMe could capture genetic signature that is highly correlated with opioid use disorder, we could show that this approach could serve as a complementary strategy for understanding genetic risks for opioid use disorder for a fraction of a cost. So we performed a GWAS of these traits and identified two genome-wide significant loci. And if we zoom in one of these top loci, we can see that the strongest signal was with variants in the gene KDM4A, which intriguingly, this gene was recently associated with clinically ascertained opioid use disorder in an independent study. In this busy spiderweb, we can see that this gene, KDM4A, on the far left, interacted with multiple compounds as shown by the number of green lines in this diagram, including serotonin reuptake inhibitors such as disulfiram, citalopram, as well as dopaminergic agents, which are drugs that are often prescribed for mood disorders primarily, which in turn are known to co-occur with opioid use disorder. Even more important was the fact that this trait shows strong genetic correlations with the largest available GWAS of opioid use disorders. And we also identified strong genetic correlations with over 200 other outcomes, which are here represented as dots, particularly other substance use traits, pain medication, as well as associations with risky behaviors. So admittedly, At that point, we were concerned that maybe we were just picking up on signal that had little to do with opioid misuse, but more to do with risky behaviors, such as that people that may be more inclined to misuse opioids may also be more inclined towards risky behavior in general. So we used statistical tools to condition our signal, here in gray, on risk in red, and it was reassuring to see that this had little impacts on the genetic correlations that we identified in the earlier plots, and here I am sampling some of the most prominent ones, particularly opioid use disorder here at the very top, suggesting that the signal that we captured is primarily specific to opioids and not merely risk-taking. So altogether, this set of results demonstrate that simple questions, like the one we studied, which capture how people misuse opioids, can help us elucidate the genetic basis of opioid use disorders. And notably, our study shows that this is possible in cohorts like 23andMe, where they have low levels or relatively low levels of opioid use disorder. Encouraged by these findings, in collaboration with Dr. Abraham Palma and 23andMe, we have launched what we've called the Prescription Opioid Genetics Study in a cohort that will be the size of UK Biobank across multiple ancestries. This survey has already been deployed, and we are in the process of collecting rich opioid, substance, and psychiatric data from well-validated questionnaires in a cohort where everyone will have tried prescription opioids at least once in their lifestyle, as we can see in this slide. We also aim to collect data on pain, trauma, and other conditions that are known to intercept with opioids. So this survey is intended to further our understanding of opioid use disorder, as well as understand the intricate relationships between prescription opioid use and misuse and the relationship with mental and physical health. We hope to complete this data set by 2024, and we would love if you could be one in 500,000. This project has not yet received any funding. If you're listening and are interested in collaborating, either by participating in the actual study, if you are a 23andMe user, or by sponsoring this project, if you are a donor, on behalf of myself and our army of scientists, we would love to work with you toward this common goal of eradicating this epidemic that has touched the lives of many. The fourth study that I would like to present today does not focus on any particular drug, but rather it uses another continuous phenotype, impulsivity. The textbook definition of impulsivity is thoughts or actions that are poorly conceived, prematurely expressed, and that often result in undesirable consequences. This is a broad definition, and in fact, we know that impulsivity is multifaceted, can be captured in many different ways. For example, by the inability to delay rewards over time, the inability to stop a response before it is initiated or after it is initiated. And each of these domains have been extremely well characterized in the field of neuroscience. This trait is attractive because it is associated with many neuropsychiatric disorders, but particularly substance use disorders, modulating multiple stages of vulnerability, including the transition from regular to harmful use to the precipitation of relapse. And so by studying the fundamental nature of this process, which is present in each one of us at a higher or lower level, we can in turn learn about the etiology of substance use disorders and other co-occurring conditions. So as part of our collaboration with 23andMe, our survey also included several well-established questionnaires that each captured different facets of impulsivity. We included the Impulsive Behavior Scale, or APS, which measures 5 different dimensions of impulsivity. And one example of this scale is, I quite enjoy taking risks. We also use the Barrett Impulsiveness Scale, or BIS, which measures 3 different dimensions of impulsivity. And one extract is, I do things without thinking. And now you can do a quick test to see what would be your answer to that question, whether you are impulsive or not. So we collected 150,000 responses on these 8 impulsive personality traits, and we performed independent GWAS of each of these dimensions. We learned that impulsivity is heritable, which means that the extent to which we are more or less impulsive, or the way that we responded to the previous question, can be attributed to genetic factors. But let's pay attention to this number, 10%. It is important to note that genes are not deterministic. Only 10% of the traits that we studied could be explained by genetic factors, which suggests that even though we as geneticists are interested in finding biological causes that contribute to behavior, the environment plays an equally or perhaps even more important role in shaping how we feel and how we act. We also learned that impulsivity traits are genetically correlated, shown by the colorful red bubbles, but that the overlap was incomplete, emphasizing what it had been known in the field of neuroscience for a long time, that each impulsivity domain is governed by different biological mechanisms. These impulsivity traits were also genetically correlated with substance use traits, as shown by this overwhelming number of red dots or positive genetic correlations for broadly all impulsivity facets, with a few exceptions, from traits spanning to alcohol, to nicotine dependence, to cannabis and opioid use disorders. And now the challenge, of course, remains for future studies to disentangle the nature of this complex web of correlations. Here's the summary of the GWAS analysis for all the 8 impulsivity traits painted using different colors, which identified 16 genomic regions associated with impulsivity. Before I jump onto the significant findings, that is, those that are above the significant threshold red line, I'd also like to note at least a couple of nominal associations that lay beneath this line. One is near the gene dopamine DRD2. This gene is responsible for regulating how our brains respond to dopamine, and dopamine is a neurotransmitter that plays a crucial role in how we experience pleasure, motivation, reward. Another one is in this complicated region, including the corticotropin gene, well known for its association with stress, depression, anxiety. Rocketing back up the top of this Manhattan plot, as I had promised, the clearer, most consistent signal was on chromosome 3, near the gene cell adhesion molecule 2, or CADM2. This gene is fascinating because it is crucial for synapse formation and plasticity, and it is enriched in the frontal cortex and striatum, which are regions that regulate reward and inhibitory processes. We and others have robustly independently implicated this gene in traits that seem to underlie deficits in inhibition, and here's the frustration that we all geneticists share, which is GWAS can only point to regions in the genome that are associated with a trait. These are not causal. We have relatively little information on whether the SNPs affect genes and gene expression, and if they do, what is the directionality of effects? To put it differently, even though we and others have robustly established CADM2 as a candidate gene for impulsivity, we still had little understanding about the mechanisms through which this gene influence behavior. So to this aim, and with the generous support of the Brain and Behavior Research Foundation and donors from Families for Borderline Personality Disorder Research, we produced CADM2 mutant mice, and we tested them in a broad battery of behavioral tasks that included measures of impulsivity. And of course, the advantage of studying dimensional phenotypes in humans is that it is easier to choose appropriate models in animals. So here we use the mouse Iowa gambling task, which, brushing over the details, measures risky responding in mice. We found that mice lacking one copy of this gene, CADM2, on the right in green, showed less risky behavior. We also measured other facets of impulsivity, as oversimplified in this cartoon, via the well-established 5-choice zero reaction time task. And again, as we can see on the right in green, loss of CADM2 resulted in less impulsive responding in this task. Beyond impulsivity measures, we did not observe deficits in any of the 2 most well-established tasks to measure anxiety-like behavior in mice, showing that this gene seems to be quite specific to impulsivity and not general aspects of behavior. And we also identified that some measures that were associated in humans, such as alcohol consumption, did not replicate in mice, but whether we're modeling the right phenotype in mice, it is unclear. But in any case, we have now validated the human loci, but more importantly, we have generated a reagent, the CADM2 mice, to study the role of this gene on behavior at the molecular, cellular, and circuit level, and we are collaborating with researchers from multiple institutions for a more in-depth mechanistic interrogation. I would like to close this chapter with another shower of praise on the use of dimensional phenotypes, such as impulsivity, measured in the general population. It is very important to note that dimensional phenotypes like impulsivity are not meant to replace clinical diagnosis, but instead they may allow us to dissect substance use disorders, enable translational research, and in doing so provide a more granular biological understanding of substance use disorders, which will be critical in extracting biological insights from GWAS in the years to come. To our knowledge, we have compiled the first large-scale GWAS datasets on impulsivity, so please do reach out if you'd like to use this data. We have reached the final project of my talk. The previous examples focused on multiple stages of substance use vulnerability, from consumption to misuse to impulsivity. My final chapter will end at the most severe stages of vulnerability for a particular substance, tobacco use disorder, using a very novel way of phenotyping that leverages passive data collection via electronic health records, that is, the information that physicians capture every single time that we go to the clinic or to the hospital. But electronic health records are not designed for research, so I must be honest, we weren't sure when we began the journey what the quality of the phenotype would be. But I hope that by the end of this presentation, you will be as surprised and pleased as we were with the set of results that you're about to see. The field of tobacco has experienced one of the greatest successes in the field of addiction genetics, particularly compared to other substances. GWAS have uncovered hundreds of associations, especially for aspects of smoking initiation, consumption, as shown in this landmark paper by the GSCAN Consortium in 2018, and further saturating the number of loci by using millions of diverse individuals in a more recent study. But later stages of vulnerability, such as tobacco use disorder, they have been largely unexplored, which is, we have established, unsurprising, because ascertaining high volumes of cases is expensive, slow, requires clinicians to establish a diagnosis using the gold standard DSM manual of mental disorders. In 2020, our colleagues from the Indigo Consortium took a different approach and published another influential paper looking into nicotine dependence via 6-item self-reported questionnaire, as shown in this slide, commonly referred to as FTND. And despite all these terrific undertakings, it was clear that as we learned from alcohol and opioid use disorders, much larger and more diverse samples would be needed to move the field forward. Over recent years, as I have alluded to, there has been a shift in the field in which electronic health records, or EHR, can be paired with genomic information to fuel genetic studies of mental health traits, which is the premise of the Psychiatric Consortium, and particularly the substance use disorder work group that I am fortunate to co-chair. So we began with a clear goal in mind. Can we prove or disprove that we can use EHR information to identify cases and controls for a trait that is otherwise hard to collect at a large scale, such as tobacco use disorder diagnosis? We studied a very simple phenotype that can be extracted from EHR, whether someone had 2 or more diagnostic codes for tobacco use disorder, more commonly known as P codes. These codes may be derived by psychiatrists, but they may also be derived simply for billing purposes, so they are imperfect in nature. And we wondered if we could show that this trait, which can be considered minimal phenotyping, if we can show that it captures signal that is relevant to tobacco use disorder, we could show that it could serve as another complementary, cost-effective strategy for genetic studies of tobacco use disorder. So we gathered data across 4 sites from the Psychiatric network across multiple ancestries, and within just 4 months, which is remarkable and one of the strengths of working in this consortium, we performed GWASs of tobacco use disorder ficotes at each of the sites. Reassuringly, tobacco use disorder ficotes were genetically correlated across sites, which encouraged us to combine the data. And here are the GWAS results for each of the 3 ancestries that we studied. We identified 55 loci in the European cohort in red, 2 loci in the African cohort in blue, and despite our attempts at increasing diversity, we did not identify any significant association in the relatively smaller Latin American cohort. So to boost power, we combined the data across these 3 ancestries, and we were positively surprised by the number of loci or dots that surpassed the significant threshold line. And here was our turning point when we realized that all the GWAS loci that we identified replicated previously known associations with smoking phenotypes, from aspects of initiation to consumption to cessation to dependence, which was the clearest evidence of the value captured by tobacco use disorder ficotes. This was also reflected not only by the quantity, but also the quality of the signals, as you will see in the next 4 slides. First, nicotine receptor genes, which of course need no introduction. Second, all drugs of abuse activate the dopamine reward pathway, and we can see here that several dopamine-related genes were associated with tobacco use disorder. Third, substance use disorders have been speculated to be a disorder of learning and memory, and we found associations with variants in a gene that encodes for a specific glutamate receptor, and glutamate is an important neurotransmitter for mediating reward-related learning and memory. And similarly, we identified variants in the gene BDNF, which is also involved in memory consolidation processes. We then aggregated all these variants and we mapped them onto genes via various methods that I will not go into the details, which identified over 300 genes associated with tobacco use disorder, which can of course now open up novel hypotheses to be tested experimentally. In aggregate, these genes were enriched in nicotinic and dopamine-related pathways, as well as enriched in brain tissues, including regions such as the frontal cortex, the limbic system, which have been studied for decades in the field of addiction, and which gives us additional clues about the specific locations or mechanisms in the brain where the effects of these genes are more likely to occur. In this busy plot, we learned that tobacco use disorder risk genes, here squished on the far right, showed interactions with medications such as varenicline, as we expected to find because it is the FDA-approved drug for smoking cessation. But we also found associations with other compounds that seem to mirror conditions that are highly prevalent with tobacco use disorder, such as the target of antipsychotics, antidepressants, dopaminergic agents. Tobacco use disorder was also genetically correlated with other comorbid conditions, particularly substance use traits, here clustering on the upper left corner, as we had anticipated, as well as other psychiatric and even medical conditions. We then extended this analysis, which used data from other public available GWAS, to another site from the psychiatric network, the Mayo Clinic. For each individual in this biobank, we calculated what is known as polygenic scores for tobacco use disorder, which broadly summarize the genetic liability for tobacco use disorder in a single score. And we tested how did those scores associated with other medical phenotypes. And this is an analysis that we call genome-wide, because we're testing hundreds to thousands of traits, association analysis, or PHIWAS. As we expected, tobacco use disorder polygenic scores were associated with the manifestation of having a tobacco use disorder, as defined via ficotes, in this independent site, which served as our positive control. But we also identified associations with a plethora of other conditions, from other substance and psychiatric disorders, metabolic, respiratory conditions, pain, among many, many others that I don't expect you to read them all, but just to show the far-reaching role that genetic liability for tobacco use disorder has on human health. We also extended this analysis to a deeply characterized sample recruited specifically for genetic studies of substance use disorders, the YELPEN sample. And again, for each individual in this sample, we calculated polygenic scores for tobacco use disorder and performed a FIWAS, in this case against hundreds of other traits that were derived from a comprehensive psychiatric interview. We were extremely pleased to see that our trait, defined via EHR, was strongly associated with many substance use traits, including nicotine dependence, here highlighted in yellow, in both the European cohort and the African YELPEN cohort, which were traits that again were defined via comprehensive clinical diagnosis. So this was extremely encouraging. At this point, though, some of you may rightly be thinking, how dissimilar really are these results from those of prior nicotine GWAS, such as that of FTND, as you may recall from an earlier slide. When we compared our results here in red from those of FTND in gray, we identified that polygenic scores for tobacco use disorder were more strongly associated across virtually all domains, including nicotine dependence, than FTND polygenic scores were. Again, emphasizing the value of collecting information on later stages of vulnerability or more severe phenotypes, such as tobacco use disorder. And lastly, we extended our polygenic analysis to a younger population, the adolescent brain cognitive development cohort. Luckily, substance use could not be captured in this very young population, at least at the particular wave that we studied in children of 9 to 11 years of age. But externalizing behaviors, which emerge in childhood and are strong correlates of substance use, they can be measured. And we found that tobacco use disorder polygenic scores were significantly associated with externalizing, here at the very top, as well as internalizing, cognitive, weight-related phenotypes. And I'd like to emphasize that these children have not been exposed to tobacco. So we may speculate that any of the associations that we observed may not be a consequence of smoking, but rather it may underlie overlapping genetic architecture across the traits that we studied. So overall, it is undeniable, after scrutinizing the data, the quantity and quality of GWAS signals in diverse populations, the enrichment in relevant pathways and tissues, the genetic overlap with nicotine-related traits from deeply characterized samples, they all provide the proof of principle that electronic health records can serve as complementary tools to study tobacco use disorder genetics in a cost-effective manner. And this work has two immediate direct implications for all of us. The first one emphasizes the importance that doctors and patients, that is, each one of us, have in advancing genetic studies of substance use disorders. None of this work would have been possible without the contribution of physicians who diligently collected the data as part of their care, or patients who consented to give their data for research. The second one questions how we approach the diagnosis and treatment of substance use disorders. The pervasive genetic overlap between tobacco use disorder, for example, and other psychiatric and even medical conditions suggests that current diagnostic mechanisms, such as the book that I have repeatedly flashed on the screen today, the DSM, it may be insufficient in capturing the complexity and co-occurrence of these conditions at the biological level. After all, as it is said, the genes have not read the DSM. So looking ahead, there's much more than we hope and we should accomplish. It is undeniable, we will all agree now, that GWAS have been tremendously successful. With the availability of newer, larger scale data sets such as all of us, or other academic initiatives such as the Psychiatric Consortium, with access to longitudinal data from millions of patients, it is clear that the number of loci associated with substance use disorders will continue to grow exponentially. But is increasing sample sizes with the hope to identify more loci all we should be doing? And when we do, what are we going to do with all the discovered loci? In other words, how are we going to turn these pockets of statistical signal into biological knowledge? If we can break this wall, it is possible that we will find better targets that could have treatment and prevention value. And we are indeed actively working on developing new methods that do not only work at the single gene level, for example, such as in the case of the CDM2 project, but that they also work at the polygenic level, even at the single cell level, so that we can have a high 3D resolution map of where and when do these genes have their effect in the brain. But we must increase diversity. The vast majority of the studies that I have presented today are dominated by individuals of European ancestry, which clearly does not represent the majority of the human population. It is many of us, and here I am only listing a few, that are working on translating and integrating research findings across ancestries and ensure that we do equitable science. Although we're only the bear on top of the iceberg, it is very exciting to see how the field will be moving forward with a goal to translate some of the most promising findings to the clinic and eradicate these devastating conditions. The work that I have presented today is the result of many years and inspiring colleagues. I'd like to close by listing anyone that contributed to this research, the biobanks, and the many smiles behind this work, many more that I could not fit in a single slide. But most importantly, the Brain and Behavior Research Foundation for launching my career and that of many other junior scientists, and together contribute to the common goal of understanding mental health and improving the lives of many. I include my email and welcome any questions or comments that you may have. This was a true joy. Thank you so much for having me.
Sandra, thank you so much for the work you're doing, and for just, this was a complicated topic, and being able to present it in a way that is as lay-friendly and user-friendly as possible. So thank you for that. We have a number of questions, and I want to go to what you spoke a little bit about, which is that genetic/environmental interaction. Could you talk a little bit about, how those two play against each other or with each other to bring about a certain phenotype?
This is such an important question, and I'm very glad that this was your very first question, because it's an important one to answer. Genes, we have shown, are very powerful ways to understand the biology of these conditions. If we can understand the biology, we can find new diagnosis, treatment, and prevention mechanisms. But genes are not deterministic, and that is very important to really come across. Genes are a determinant, but they really play a major role with the environment. Someone may have a high genetic liability for substance use disorder and yet be in an extremely protective environment and never manifest the genetic liability. So it is very important and encouraging for anyone that is listening that the environment and the support that we can give to those with substance use disorder is incredibly valuable.
Very good point, very important point. Where do you see the work going in terms of based on the science, developing new clinical interventions? What do you see happening? What do you hope might happen in the next 5 to 10 to 20 years with regards to this?
Yes, thank you, Jeff. So I I read some of the questions that some of the attendees had an hour or so before the presentation, and one of the points, or many of the points that they raised, was exactly that. So how can we transform what I have discussed today into something meaningful that can affect people's lives? And so here I have some additional slides. To answer that question, perhaps we need to go back to 2015, when Barack Obama presented this exact vision, the Precision Medicine Initiative. This initiative aspired to take into account the individual variability in genes, and we can see him here, Barack Obama, pointing to the double helix, to improve healthcare. And how could we do that? And why should we do that? So if we want to realize this office in 2014, for example, 2040, or even a visit to the doctor from our own devices at home, the question that I'm gathering is, how far away from that? So we could use genetic information to improve diagnosis. And what genetic studies have taught us is that diagnostic categories are dynamic. The boundaries within disorders are paper thin. And in fact, one of the biggest revelations has been that these genes or loci are shared across psychiatric and even somatic disorders. And so what I think will be happening in the next years, it will be taking into account this information that we are learning through biology to suggest new ways to define and potentially treat substance use disorders. We could also use this genetic information, for example, in the form of polygenic scores, to delineate paths of risk, but also resilience. And again, it's important to know it is just paths of risk. It's not deterministic. Just hints to indicate how genetic variability may interact with everyone's environment and lifestyle. We could also use genetic information to predict how substance disorder may progress, and this is particularly important in children and adolescents when they have not been exposed to substances, but they may in their teens. And we could also use all these amounts of data that we're gathering from GWAS to identify genes that could serve as actionable targets. And we have clues from other disorders, particularly from more of the medical field, diabetes, that have proved to be incredibly helpful. And this is important because, for instance, now there's only FDA-approved medications for treating alcohol use disorders, and these are still very limited. So we may see some progress in this field. And we can also use genetic information to help identify or offer better treatments. So for example, matching patients to specific treatments and reduce trial and error. And perhaps more importantly, to develop therapies that treat the underlying causes. Again, we have seen that comorbidity with other substances and other psychiatric and medical conditions is the rule. And so it will be very important to know how can we treat all these conditions.
Excellent. Yeah, go ahead.
I just wanted to add that if we really want to incorporate all of this information for especially prevention, when we go to the doctor and making the decisions of diagnosis, prevention, and treatments, it is also very important to note that at this moment in time, genetics can only move the needle by little bits. Genetic information can only predict traits in a very modest way. So it will be very important to know that if we want to realize this vision, we will need to create guides for clinical implementation when and if we're ready, and educate the public on how to interpret genetic risk.
All excellent points. And just sort of, I was thinking along these lines as before you said it, but has there been studies that look at differences between people who are able to achieve recovery from chemical dependency versus those who may have more difficulty doing so?
That's a wonderful place that I want to go. At the genetic level, such studies, we're in fact in the process of conducting this study, which is looking at resilience. Resilience is very powerful because it can give us clues about the environment protective effects of the environment or other personal characteristics that may place people protected to substance use disorder. But I'd like to note something, and it is very important that I have the opportunity to have everyone here, and thank you to all that stay until the end. If you are struggling with this condition, or you have a family member or a friend, and if you think, how can I help genetic studies? It is very important that we increase participation in research. And that's why the focus of my talk, I wanted to really emphasize study designs and the importance of involving everyone in research. The question that you raised, Jeff, is very important, but it will be very hard to find people that meet these characteristics. So it is really my desire that if there is any take-home message from this presentation, is to empower everyone to be involved in science. These studies are very costly, not only in terms of funding, but also because it is incredibly challenging to reach such large sample sizes. So we need the help from everyone. And I have shown that it is not necessary to only just study extreme ends of the spectrum. We can also learn a lot about learning about personal characteristics, such as how impulsive we are, or how we react to opioids when we try them for the first time. And we can learn a great deal about substance use disorders by learning about these more normative behaviors, if you will.
Well, Sandra, I want to thank you again for taking the time to do this outstanding presentation and for the work that you've been doing. Your passion for this comes across so strongly, and it is really inspiring. So thank you for all that you do. And we're looking forward to hearing updates from you in the not too distant future as you get more information from this work. So thank you so much.
Thank you, Jeff.
I'm pleased to share that BBRF has received a $1 million challenge match for 2023 from two very generous family foundations that are passionate about our mission. The goal of this match is to accelerate the momentum in brain research as further breakthroughs are still needed. You can participate in this initiative if you're a new BBRF donor, a past donor who lapsed but makes a new donation this year, or a current donor who increases your 2023 gift, with your increased amount to be matched. I hope you'll consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operation expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientists. To make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it with family or friends, please visit the events and webinar page on our website. And please join us again on August 8th at 2 p.m. Eastern Time, when Dr. Emily Ricketts, Assistant Clinical Professor in the Department of Psychiatry and Biobehavioral Sciences at the University of California, Los Angeles, will present Morning Light Therapy for Tourette's Disorder. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and substance abuse and enable more people to live full, happy, and productive lives. Thank you.